Hello everyone and welcome to the first series of revision notes about cell theory, which is 1.1 in the IB biology syllabus. There are three main parts in cell theory. The first part being that cells are the basic unit of life. Now, what does this mean? A cell comprises of a series of organelles within it, such as a nucleus. However, a cell itself is alive, a nucleus is not. This means that a cell is this most basic unit. A second section of cell theory is that all living things are composed of cells. This means that anything that is alive in the world that we live in must have cells within it. So something like a virus would not be considered alive as it does not contain these cells. The third key point from cell theory is that all cells come from pre-existing cells. We know that cells are derived from reproduction, whether that being asexual, so things like binary fission or mitosis, or through sexual, which of course is your meiosis. Cells cannot be spontaneously generated. They must come from a central point of origin, with this being your parent cell. A useful experiment within this course is by that of Louis Pasteur, who disproved the idea that cells could grow spontaneously. In his experiment, he had two different flasks, one of which was sealed and one of which was open to the external environment, and he observed that the one that was open had microbial growth in it, whilst the sealed flask had no growth. This therefore proves that cells cannot come about spontaneously in an enclosed environment. There are a multitude of different functions and characteristics of cells. The first one notably being that cells have a cellular structure. According to cell theory, all living things are composed of cells, and within those cells there is a structure of organelles, whether that being membrane bound in your eukaryotic cells or not membrane bound in your prokaryotic cells. Some examples of structures within cells are your nucleus within your eukaryotic cells, your Golgi apparatus, and your plasma membrane, with a cell wall also being present in plant cells. A second characteristic of all cells is their ability to respond to their environment. Cells have receptors in the plasma membrane, which allows the sending of signals into the internals of the cell to allow a response accordingly. This allows regulation within the cell, and this allows cell stability in the event where an environment changes its ability to function. A third characteristic of cells is their ability to grow and develop over time. We'll talk about later this idea of a surface area to volume ratio of cells. However, we know that cells generally divide and pass on their genetic material to their offspring, ultimately allowing a development over time and a growth from the parent organism. An example of growth within an organism is metamorphosis. We know with caterpillars transforming into butterflies, this is a physical change, and it's an example of how organisms can grow and develop over time along with cells. It's important to note hormones, which are your chemical messengers, are things which allow this change within cells to occur at a particular point in time through messaging, so to speak, the internal compartments of a cell to allow this change to take place. An example of catabolism within a cell is that of cellular respiration, which is, as a simplification, a breakdown of glucose to form ATP. So it's this breakdown of a complex molecule to form small molecules along with its energy. An example of an anabolic reaction, metabolism, is photosynthesis, which is the involvement of carbon dioxide and water, which are your small molecules, to synthesize this larger molecule of glucose, which is also with usage of sunlight. However, ultimately we can get this picture that there are small molecules which synthesize this large molecule, and for catabolism, a large molecule synthesizing a small molecule and energy. It's important to note that enzymes play an important role in metabolism through their ability to catalyze a reaction by lowering its activation energy. An enzyme is something seen very commonly throughout humans, and it allows metabolism to occur at a catalyzed rate. Without the presence of enzymes, we know that metabolism would be extremely difficult due to the time it would take for such reactions to occur. An essential characteristic of all cells is having a metabolism, which is the series of countless chemical reactions within a cell. There are two forms of metabolism, both of which occur within our bodies. Catabolism is the breakdown of a larger molecule into the products of energy, along with smaller molecules, which I'll signify as SM. Anabolism is effectively the opposite, with energy and small molecules coming together to form this larger molecule. It's important to note that this energy can be in different forms. However, it's most commonly seen as ATP, which is your adenosine triphosphate, and it's a common energy source, notably in humans. Homeostasis is another characteristic present within cells. Homeostasis is the maintenance of the internal environment and keeping it at a certain range which is suitable for cellular function. Homeostasis can be seen in keeping a balance of water or minerals at a certain level which is optimal for a cell to function. We know things like osmosis and membrane-based transport allows homeostasis through regulating, for osmosis specifically, regulating this water balance within a cell through the semi-permeable membrane, and through your membrane transport, whether it be active or passive, allowing a regulation of certain elements, an example being your sodium potassium pump, which will be discussed later throughout this revision course. Reproduction is an essential part within cells, this being either sexual or asexual, as discussed previously. Sexual is amongst two parents, involving the fusion of your haploid cells, with asexual production being one parent and resulting in genetically identical offspring. Asexual can be seen in mitosis along with binary fission, whilst sexual we know is our meiosis. The next characteristic of cells is their ability to exchange matter with their environment. This can be seen in excretion from cells through metabolic waste. It can also be seen through other forms such as heat loss from organisms such as mammals. Organisms and cells also consume resources from their environment, and this stems to the fact that they are exchanging energy. We'll now move on to some exceptions to cell theory, with the first one being giant algae. As depicted in this illustration, giant algae looks something along the lines of this, with it being especially large for a unicellular organism, up to 10 centimeters. The discrepancy of this giant algae is that it is large. Typically, cells are on the microscopic level and they're not something that we can see with our human eyes. However, giant algae is a notable exception, being much larger in size than your typical unicellular organism or cells in general that are a part of an organism. A second notable exception to cell theory are muscle cells, which are seen in the human body. As illustrated here, muscle cells have the presence of multiple nuclei, with each line here representing a muscle fiber and the black dots representing nuclei. This is a discrepancy against cell theory, as eukaryotic cells, such as these cells, typically only contain one nucleus. However, muscle cells have a notable exception containing multiple, going against the idea that cells are typically autonomous with multiple nuclei, meaning that there are separate points of control, so to speak, in terms of the genetic material across the whole organism. Seen in the course quite commonly is paramecium, so it's important to be aware of its structure and its overall function. We know paramecium is unicellular and eukaryotic, living in a freshwater environment. We know it's a heterotroph, which means it relies on other organisms for energy. And we also know that paramecium is able to carry through all life functions. Here is a simplified diagram of paramecium. We can see that the cilia on the outside can be used to assist cellular movement and locomotion. We can also see the oral groove and anal pore, which shows paramecium's use for ingestion and excretion of food, so that interaction with the environment. We can see the contractile vacuoles as well, which allow that water balance within the cell, and ultimately can allow that regulation within the cell, which of course we know as homeostasis. Notably as well, the food vacuole allows this ingestion of food, and in turn with that oral groove and anal pore, we can see that paramecium can successfully ingest and acquire nutrients from other organisms, with it being, of course, heterotroph. Surface area to volume ratio is the next concept that will be discussed, and it is relevant across all cells, with the surface area representing the membrane around the cell, whilst the volume representing all of that internal region of the cell. As the volume increases more rapidly, as the volume increases, it's more rapid than your surface area increasing. So this therefore means that cell division will have to occur to regulate this, because as a cell's volume increases, there is a larger volume of metabolic reactions occurring. This therefore means that there's more nutrition required to sustain the cell. However, for a smaller cell, there is less metabolic nutrients and waste required, but there is also a higher level of surface area which allows a high level of diffusion to occur and a faster, more efficient amount of transport amongst the cellular components within and moving in and out of the cell. So as a cell grows larger to this point here, it can no longer serve the requirements of the cell due to the volume being too large and the surface area not being able to support it to sustain the cell. Decreasing a surface area to volume ratio will ultimately simulate cell division, and this will be through mitosis or binary fission. And through dividing, such as this big cell here, through dividing into two smaller cells, we can see that the surface area to volume ratio is now balanced, ultimately allowing more cell efficiency, allowing that faster level of diffusion, and ultimately allowing the nutritional requirement the cell needs to survive through having that surface area available. Some interesting cellular adaptations, if you will, to maximize this surface area in order to quickly diffuse material amongst cells can be seen in the following. Firstly, a neuron, and these are represented to be dendrites. Ultimately, these long and thin dendrites allow nerves to quickly pass information along to other nerves, with this structure allowing it to be done efficiently due to its very, very large surface area in comparison to such a small volume. It can also be noted here in the intestine, villi and microvilli are seen to allow this absorption of nutrients from the food in which we consume. Because these are so fine and small, with such a large surface area and small volume, the same concepts can occur, with the absorption of nutrients being able to occur at a fast and efficient rate for what other areas of the body require. The next section of this revision guide will focus specifically on multicellular organisms. Multicellular organisms contain a diverse range of cells, which allows more efficiency along with the formation of specialized structures. For instance, in humans, there are a variety of specialized cells, things like nerve cells or skin cells. Nonetheless, it's evident that through having multicellular organisms, there is, in a sense, teamwork amongst these different types of cells, which ultimately allows the body to be more efficient in undergoing daily activities. The question of how these cells come about is due to differentiation, which is where cells contain the same genes, however, different phenotypes are expressed such that they have a different function, potentially a different structure. Ultimately, this differentiation is on a genetic scale, and it allows cells to be more specific due to their unique expression based off their environment. And this, of course, is different within different cells. So, for instance, in a muscle cell, it may contain, let's denote gene A, B, C. A muscle cell may contain gene A and gene C, whilst a skin cell may also contain gene C, however lacks the presence of gene A and instead has gene B. Something along these lines show this differentiation and thus how different types of cells can form within multicellular organisms. Stem cells link strongly to this idea of undifferentiated cells, and they're present within humans typically in the early stages of pregnancy. So stem cells are undifferentiated and they can divide indefinitely to form more stem cells. Most notably, they can divide to form a specialized cell, meaning that if I have a stem cell, it can develop into any human bodily cell. So this stem cell could turn into a nerve cell, which I'll denote with an N, skin cell, etc. This is what makes stem cells so valuable in essence. And there are multiple phases of stem cells, if you like, as they go from no differentiation, which I'll denote as N, to a fully differentiated cell, denoted as D. So firstly, your most undifferentiated stem cell is known as toripotent. Toripotent stem cells can become any body cell, including cells in the placenta, and we find these in zygotes. The next phase, if you will, of stem cells are pluripotent, which can also become any bodily cell. These are found in your blastocyst, so we can note that they're both in your very early stages of pregnancy. The final form, and I'll just change colours for the sake of simplicity, is multipotent, which has partially differentiated. Partially is our key word here because it can still become multiple related types of cell. So although it's not as valued as your pluripotent or toripotent, multipotent stem cells are nonetheless able to differentiate into a specific type of cell, and these are found typically in the umbilical cord. Typically, multipotent cells are a lot easier to acquire. When looking at the ethics of stem cells, multipotent, being from the umbilical cord, are much easier to acquire. Something like a zygote, so a unicellular organism, if you will, a lot harder to extract, and of course the ethical implications of a zygote ultimately turning into a fetus, an embryo, and then a fetus, of course. Much harder to extract, therefore ethical concerns surrounding them. However, nonetheless, they are seen as valuable due to the potential stem cells have to treat varying conditions. Stargardt's disease is an example of a condition which can be treated using stem cells. So as a brief summary, Stargardt's disease is based in the eye, where cells within the eye, effectively in the central area, stop functioning such that people cannot view directly out from their eye. They can see from like the corners, but ultimately there's this like large circular type blank spot which cannot have vision from, if you will, for people with this condition. So stem cells can generate these healthy eye cells. We can specialize a stem cell into the particular eye cell that we need in order to replace the cells in this eye with Stargardt's disease, creating this. Stem cells can be guided into becoming specific cells to generate and repair the damaged tissue, which makes them notably effective in curing this disease. Leukemia is another real-world application for using stem cells in a therapeutic sense. Leukemia is where an abnormal production of white blood cells occurs, usually in excessive amounts. In terms of treatment involving stem cells, chemotherapy can be undergone first, which allows this removal of the abnormal white blood cells. Following this, a bone marrow transplant can be undertaken and used to replace the multipotent stem cells that form the blood cells in the bone marrow. So the use of stem cells allows the implantation of good and properly working multipotent stem cells, which in turn allows your healthy production of white blood cells for the body to successfully function. Moving on into the skill-based section of this syllabus point, a necessary skill is to calculate the magnification from a microscope. Now we can see our equation is the ocular magnification times the objective magnification. Now the ocular magnification is found from the top lens section of a microscope, whilst the objective lens is found just above where you place your slide for viewing. So as an example, let's denote the objective lens to have 4 times magnification and the ocular lens to have 10 times magnification. So when calculating, we simply just substitute in the values accordingly, so it's going to be 10 times 4, with the overall magnification therefore being 40x. So this is the first skill required for this course. A second important skill is to estimate a sample size when you're given an image of an organism or something similar under the view of a microscope. To do this, we first have to know the field of view, which is, if this is a microscope image, your diameter of such image. So as an example, let's say the diameter is 400 micrometers. The next step that we need to do is to calculate the fraction of the organism that takes, like how much space does this organism take up in terms of the field of view. So for instance, if that circle that I just drew was the organism, we can separate it into segments. So we can see there that I've just separated that into 1, 2, 3 into fifths. Roughly speaking, this organism is about 1/5 of that diameter. So therefore, what we can do is 400, which is our total diameter, times 1/5. And if we get out our calculators, 400 times 1/5. And that will equal, so our final calculation will be 80, therefore we're looking at 80 micrometers for this example. So as a summary, using the field of view, finding the fraction that the organism or similar takes up, and multiplying the field of view by that fraction is how we can calculate an estimated sample size. And that marks the end of the 1.1 syllabus point for this IB biology course. Thank you for watching along, and we hope that this revision guide will be of assistance for your upcoming exams.